Okay, now we are going to start into some kingdoms that might seem a little familiar to you, and today we're going to start talking about the animal kingdom. Since we've been talking about supergroups where protists were spread across all of them, animals are specifically in the supergroup Apasthaconta. And they share that supergroup with fungi and certain protozoans. So let's talk about what makes animals different from other eukaryotes. They are multicellular eukaryotes. That doesn't make them uniquely animals, however, of course. So when we look at multicellular eukaryotes, we divide them first based on their nutritional acquiring techniques. So we have heterotrophs, and those are the ones that have to get their energy from outside of themselves. And then our autotrophs, our self-feeders. And in multicellular eukaryotes, those are going to be our plants. So back over here to the heterotrophs, that falls into two different categories. One is the fungi kingdom, and those are saprotropes. Those are the ones that do our decomposition, and we'll talk about that a little bit later. But what we're going to focus on today is the animal kingdom. So they are multicellular eukaryotes that are heterotrophs. So those are animals. They ingest and digest their food. And then they go through a series of developmental steps to produce organisms with specialized tissues. Within the animal kingdom, we divide animals into two different categories, the vertebrates and the invertebrates. Vertebrates are those that have a spinal cord or a backbone that runs along the dorsal side, at least at some stage of their development. It doesn't have to be in the adult stage. And then the invertebrates are those that do not have that. So let's talk about, according to the theory of macroevolution, how animal cells got here. There is an additional hypothesis within that is, it assumes that the theory of macroevolution is correct. And so on top of that, we build on another hypothesis, and that is the colonial flagellate hypothesis. And that is just as it's stated here in quotes, animals are descended from a common ancestor that resemble a hollow spherical colony of flagellated cells. So these choanoflagellates are believed to be the common ancestor. So right here, the choanoflagellates were a protozoa that are single cellular, but they are eukaryotic cell. They have these collar cells a collar to them, and they have a flagella. So according to the colonial flagellate hypothesis, we begin with a single choanoflagellate that then form an aggregate together. And once they form that aggregate, they begin to make this colony that is spherical in shape. And then from that, then it was thought that due to mutations, we start to get specialized cells for reproduction, and then an infolding is created and starts to make specific tissues. So that's how the colonial flagellate hypothesis works. So there are at least 36 different phyla within the kingdom Animalia. So how do we classify them? We do that based on 6 criteria. And the first of those is the number of germ layers that they have during development. And a germ layer is simply cells that arise during embryological development. And there are 3 types. There's the ectoderm, mesoderm, and endoderm. Okay, so as we are looking at the embryonic development, we're going to look at that body plan. And if you'll notice, just to start with, this is what we call a blastula. Looks very similar to that colonial ball that we saw from the choanoflagellates and how they gather together and make that spherical shape. So that's why that hypothesis came to be. But during development, there are 3 possible germ layers that develop. There's the ectoderm, which forms the ectoskeleton or your epidermis. There's the mesoderm, and that develops into the internal organs. And then there's the endoderm, which forms the inner lining of those organs. And we start to classify our animals based on how many germ layers they have during development. So first of all, it can be diploblastic, meaning that they only have 2 germ layers, and typically that's the ectoderm and the endoderm. So those are animals that develop tissues but no specialized organs, and such as Cnidaria. The only exception to that rule is phylum Porifera that we'll get to, that has 2 germ layers but has no tissues whatsoever. And then the other way that animals develop is as triploblasts, meaning that they have all 3 of those germ layers, and ectoderm, mesoderm, and endoderm, and they develop specialized tissues. So that's how we begin to break down animals into different groups. Okay, so let's talk a little bit about our triploblasts, because that's where most of our more complex animals fall, because they have the internal organs. So triploblasts then can flow even further and be divided between what we call protostomes or deuterostomes. And that really all has to do with which develops first, the mouth or the anus. That's really all those 2 terms mean. So to begin with then, our protostomes, the mouth develops prior to the anus. So the mouth develops first. And most invertebrates are protostomes. The other option is the deuterostomes, and that means the anus develops first. All vertebrates are deuterostomes. So the way you might be able to think about that is between diploblast and triploblast, the triploblast is more complex because it has organs. Out of the triploblast, the deuterostomes are the more complex and more like higher organisms. Okay? So, I know it's a lot to sink into. Might even be helpful to draw a flow chart with that, just so that you understand where that comes from. So, both protostomes and deuterostomes develop in 3 major stages. So we're going to go through those real quickly. So whether an organism is a diploblast or a triploblast, there are 3 stages of their embryonic development. The first is cleavage, the second is blastula formation, and the third is coelom development. So let's look at those. So we're going to start with cleavage, and we're going to look at the differences between protostomes and deuterostomes, because as those embryos develop, they do develop very differently. So the definition of cleavage is just cell division without cell growth. So in protostomes, which is this top line here on our image right here, protostomes, here we have our first cell, and then it quickly divides into 2 and then 4. Cleavage happens in a spiral pattern. So you can see that as we get like up here between 16 and 32 cells, that cleavage is happening in a spiraling formation. And what's interesting to note about this is that the daughter cells are fixed and determinate. What it means by determinate is that the cells contribute to development in only one way. It means that if you take one of those cells away from the rest of them, it can only be that type of cell. It doesn't have the ability to take on different forms. Okay? In contrast to that are the deuterostomes, which are here on the bottom. Deuterostomes have what we call radial cleavage. So that just means basically if you were looking at a circle, everything divides in the same pattern every time. Okay, so you're going to have a radial cleavage. And the daughter cells in deuterostomes are what we call indeterminate. That means that if they're separated from other cells, each cell can grow into a completely new organism. So you can think of that kind of like, I'm trying to think of it. Plant cells are coming to my mind. What's the word? If you were here, you would help me. Stem cells. There we go. So you can think of that like stem cells. They are indeterminate cells, which means you can take them and they can become other types of cells. So you can grow a complete organism from one of these cells because they are indeterminate. That's why during cleavage, if this group of cells here breaks off from this other group of cells here, then we get identical twins. And that's how that happens. So that's cleavage. The second stage is blastula formation. So let's talk a little bit about definitions here. A blastula is just a hollow sphere of cells. The indentation that follows produces what's called a blastopore, and that blastopore is what develops into either the mouth or the anus, depending on if you are a protostome or a deuterostome. So let's look at this one in a little bit of detail here. So let's start with our protostome. So here's our blastula. Note that both of them start out the same way, a hollow sphere. And by the way, this is where the colonial flagellate hypothesis came from, because this blastula, being that hollow sphere of cells, looks very similar to when the choanoflagellates come together in an aggregate. So because of their similarities, that's where that hypothesis comes from. Okay, so protostomes. Once we have this blastopore indentation forming, then that portion right there is what becomes the mouth. And so because the mouth forms first, that is a protostome. Now, if we look in our deuterostomes, again, we have the blastopore right here. Okay, and then what happens is that's where the anus forms first. So wherever that blastopore turns into either a mouth or an anus, it determines whether that's a protostome or a deuterostome. Just another interesting thing to notice that they have the column here and here in the protostome, and those fill in this pocket right here. In the deuterostome, that column starts in a different place, a different location, and then fills in here as well. Slight differences. Mostly what I want you to get out of this is I want you to know what a blastula is and what the blastopore is, and then if the blastopore forms a mouth, then it is a protostome. If the blastopore forms an anus, then it is a deuterostome. The third way that we classify animals is based on their coelom development. So let's talk first, what is a coelom? That's basically the main body cavity, and it's positioned inside the body to surround the digestive tract and other organs to protect them from the outside. And a true coelom is lined completely with mesodermal cells. So we find coeloms in some protostomes, but not all, and in all deuterostomes. So you can tell that that is an evolutionary advantage. So we have more complex organisms containing a coelom. So in protostomes, when we were looking at that blastula, the mesoderm arises from the cells that are located near that blastopore, and they split and create a coelom. In deuterostomes, the way the coelom is created, there's a pair of mesodermal pouches from the wall of the primitive gut, and they enlarge until they fuse together, creating that coelom. It's not really important to know how they are created, but I just wanted you to have that information in case you were interested. But do know that we find coeloms in some protostomes and in all deuterostomes.
So then another way that we categorize our animals is based on the type of coelom they have. We've talked about what the coelom is and how it develops. And so when we are looking at our organisms, we can kind of categorize them 3 ways based on their coelom. Are they acoelomates, meaning they have no coelom? Are they pseudocoelomates, which means they either have a coelom that doesn't completely cover all their organs, or it may not be entirely lined with mesodermal cells? Or our third category is that we can have true coeloms, or a coelomate, which means that it has a complete coelom entirely lined with mesodermal cells. Okay, and then we also want to look at the body plan, the entrance and exit of food waste, so basically the digestive tract. They can either have a sac body plan, which is an incomplete digestive tract, meaning there's only one opening that is for the mouth and the anus, or it can have a tube body plan, which is a complete digestive system, meaning it has a separate mouth and separate anus. So that is another way of categorizing our animals. One thing that's an easy way to start categorizing our animals is based on their body plan, the symmetry that they have. And we can break those down into asymmetric body plans, such as sponges. If we were to cut them in any particular orientation, we wouldn't have mirrored halves, we wouldn't have any type of symmetry. So that's the first symmetry style. The second is radial symmetry. That means their bodies are organized in a circular fashion so that any longitudinal cut is going to produce 2 identical halves. And an example of this here is the sea star. If I were to cut it here, these 2 halves are going to be identical. And then there's bilateral symmetry, and that's found in humans and in this cute little seal here. If we were to cut in half, we have a defined left and right half. Usually they mirror each other. There is an anterior and posterior end to the organism, and that usually is accompanied by cephalization. Cephalization is just a fancy term that means there's a localized brain with special sensory organs. So the way that the body plan is laid out based on symmetry helps us to organize our animals. And then another way is simply the segmentation of the body. The separate segments tend to have specific functions. That's pretty recognizable here in this crayfish. We have the top part is the head. That's going to be specialized for eating. That middle section has the walking legs, that's what it's specialized for, and then the back section has swimmerets, which allows it to swim in the water. And almost all more advanced organisms have some type of segmentation to their body, and how that's organized is one way that we can categorize our animals. And then, of course, lastly, the molecular DNA. How are they similar versus diverse? Based on their molecular structure. And we won't go into details about that, but obviously groups of animals that are more closely related have more similar DNA structure, as opposed to those that are less, have less similar DNA. They tend to be thought to be more evolutionarily separate.
Okay, so I can hear it now. Y'all are saying, Professor Scott, that's a lot of information, and it is, and I totally understand that. Especially if you're not familiar with embryonic development, so I would definitely take some time to go back over those slides, read very thoroughly in your textbook about those, so you can get a handle on them. But now we're going to move into some phyla of the animal kingdom, and we're going to start with invertebrates. Just keeping in mind what you've talked about, and so when we get to our invertebrates, particularly phylum Porifera that we're about to talk about, this is where the diploblast versus triploblast start to come in. So phylum Porifera is going to be a diploblast, so it only has 2 germ layers to start with. So that's what we need to start with. So let's get into them. This is going to look mostly familiar to you because they are sponges. So the 2 phylum that we're talking about today are phylum Porifera and phylum Cnidaria. And those are exciting to me because I spent a good portion of my life absolutely loving marine biology, so this is where it starts, right? The most simple organisms, other than the protista that we looked at, in the ocean, and they're fascinating and beautiful, so we're going to start with that. All right, so Porifera are sponges, and they're the only animals that lack true tissues. They have just a few cell types. They don't have any nerves or muscles. Their body plan is a sac-like body plan. And what is meant by a sessile organism is that it can't move. So it stays put. They do have pores. We're going to talk about those, obviously in aquatic habitat. And they are filter feeders. They are also thought to be the oldest animals as far as phylogenetics go. There is some debate about maybe comb jellies, came to be earlier, again, according to the theory of macroevolution. So let's look kind of in detail at the body plan of our sponges. We said that they are filter feeders and that they have a sac body plan. So what a filter feeder has to do is it has to be able to move water through its body in order to filter out any organic matter in the water column. So the way that sponges do that is, first of all, they have these pores, and in the pores, these pores are called ostia, and the water comes in through them. So the small pores on the side are called ostia, and then the large holes at the top here are called the osculum, and that is where the water comes out. So in through the ostia, out through the osculum. Okay, so what happens within the body itself? Well, incidentally enough, there are what we call collar cells, and they should look very familiar to you because we talked about choanocytes before, and it's basically the same body plan as the individual choanocytes that we studied before. They have a collar here that have basically kind of like cilia, and then they have this flagella. And the flagella work together to move the water along and then bring it through. And the interior of the entire canal or the entire body of the sponge is lined with these collar cells. So the water comes through and is then brought into the amoeba site, which is another particular type of cell, and that's where the food is digested. Now, having said that, while we're looking at this body plan, I just want to also point out another cell type that our sponges have is what's called a spicule. It's these little yellow things here. Not all sponges have them. Some do. The ones that do, spicules are sometimes made out of silica. They're meant for defense. So they're very pointy, so if something were to try to eat the sponge, they would get poked in the mouth and not want to do that. So that's our body plan of our sponge. So here's a little bit better image of the spicule that we were talking about right here. So all sponges are made up of a particular protein called spongin, and that makes up these tough fibers. That's what gives it its rigidity. It allows it to have support. So all sponges have spongin. Only some have spicules, and those are the ones that were colored yellow in the drawing on the previous slide. And they are mostly made out of silica. So you can imagine if you have these sharp glass pieces that animals are not going to want to bite them. So the type of skeletal structure that is seen in our sponges are what we use to classify our sponges. So we have calcareous, which have spicules that are made of calcium carbonate or like chalk. We have siliceous, which is spicules made of silica or glass. That's this right here, and you'll get to see that in lab. I will tell you when you are in lab, don't touch it because it's pretty much just like fiberglass, and you don't want to get that in your fingers. And then, of course, the third would be spongin, which has no spicules, just has the spongin fibers. And that's primarily what gets used commercially speaking. If you've ever seen these in the grocery store or in like Bath and Body Works or something like that, sponges get used just for bath sponges. They get used in ceramics. If you've ever taken a ceramics course, once you have your clay, you wipe it down with sponges to kind of smooth out the surfaces. It can be used for painting or furniture refinishing. They have all kinds of uses. The nice thing about sponges is that you can break off a piece and they have regenerative qualities, so they can reproduce more and more. Having said that, most sponges today are synthetic. If you do get a natural sponge, it is a huge difference. So it is nice to have. Just it's a matter of conservation. So how much do you take? How much do you leave? That sort of thing. So that was phylum Porifera. Now we're going to move into another phylum called phylum Cnidaria. And again, this is still invertebrates. So let's look at some of the characteristics of the Cnidaria. You'll see some examples of them on the left. The top one is what we call a hydra. And then, here we go, right there, that's a hydra. And then your sea anemones, and then your jellyfish. These are all Cnidaria. Typically, they're either tubular or bell-shaped. They like to live in shallow coastal waters because they need the salinity, but they also like it warm. So you see a lot of these in the tropics. They do have a radial body plan. They are specifically named for the specialized stinging cells they have called cnidocytes. So we'll go into that a little bit more too. Their body has 2 layers, but it is still a sac body plan. And there are 2 different forms of Cnidaria. There's the polyp form and the medusa form. So let's talk about these cnidocytes. Cnidocytes are specialized stinging cells that contain what we call nematocysts. And if you look in this image here, oh, let me get my pen, sorry. This section right here is the nematocyst. And that's just a fluid-filled sac, and within that sac, there's this long coiled thread. So you can see it coiled up right here. And it is under a lot of pneumatic pressure. So it's like taking something and like maybe even a balloon and turning it inside out and making sure that it's blown up, but just holding it there. That's what the nematocyst is like. Once it gets triggered, then it gets put back in the right direction and it's flung out very, very fast. And this right here can be used to trap its prey, or it can sometimes be used to inject toxins into prey. I did include, I believe it's just a sheet on the Sakai site in your extras that explains that a little bit more. It's really quite impressive. Okay, so we said that the Cnidarian had a 2-layered sac. So the inner layer of the sac is what secretes the digestive juices. It's that internal cavity. And so that serves as its gastrovascular cavity because it doesn't have specialized tissues for that. And it also acts as a hydrostatic skeleton. So the amount of water that's in it is what gives it its rigidity. And it's derived from the endoderm, just like you would expect for the inner layer. Remember, these are diploblasts. The outer layer would then, of course, be derived from the ectoderm, and that's what gives its protective epidermis. So we said that their body form comes in two different forms. The first is the polyp form, and that's when the mouth is directed upward, such as in Hydra or in coral. So I don't know if you knew that coral was a Cnidaria. It just has it excretes calcium carbonate that's nice and hard to house it, but inside it's this very soft Hydra type body plan with the mouth directed upward. So our Hydra is right here. That's what that looks like. So every bit of coral is like this as well. So that's the polyp body form. The Medusa body form is like this. These are our jellyfish, is what we usually call them. And the mouth is directed downward. And this form has what's called mesoglia. And that's what fills this whole cap right here. And it's like a jelly-like substance that's between the endoderm and ectoderm. And then there are polyps that form from that. So those are the two body forms. So we have two different classes of cnidarians. The first class is the hydrozoa, and the second class is the scyphozoa. So the hydrozoas are mostly the polyp type stage. They do have a short medusa stage when they're reproducing. And often they live in colonies where specialized polyps perform particular functions. So examples of this are the hydra, and we're gonna see these in lab. In fact, we'll probably get to see all of these in lab, but we will particularly be looking at hydra. And if you notice right here, there's this little bud that forms. That is its form of asexual reproduction. It can also reproduce sexually, and that's when you start to see the medusa stage. And we'll definitely take a look at that in lab. What's also included in the hydrozoa are Portuguese man-o'-war, which are very large, and they live in marine environments, and it's a colonial group, like a colonial, what am I trying to say, life form. And we'll look at that as well. And then the second class is the scyphozoa, and that's the jellyfish. And they live predominantly in the medusa stage with a very short polyp stage. So let's look at hydra, because I want to go into detail with that. They are kind of the typical cnidarian. That's what, usually in particular phyla we have one organism that we go, oh yeah, that's the one that like most represents that particular phyla. So hydra is it for the cnidarians. So they have what's called a nerve net. So it's a very simple network of nerves that literally looks like a net. And as I pointed out before, they reproduce asexually by forming buds most of the time. They can also reproduce sexually. And they have wonderful regenerative properties. And that's because the cells in the epidermis are capable of reproducing the entire organism. Remember, that's what we call indeterminate. So it's of great interest to medical doctors as to how the epidermal layer can do this, because we would very much like for us to be able to do that with human tissue, just so we can have regenerative properties. So this is our picture of the hydra, and let's look at that in a little bit of detail since we got to do that with our phylum Porifera and looking at our sponges. Let's take a look at our Hydra. That's our representative for our Cnidaria. Keep in mind that the Porifera sponges had no nerve tissue, none whatsoever. So it's a slight advancement when we get to the Cnidaria because now we have a nerve net, just very simple all along. And here is our two layers, our endodermis and our ectodermis, and there is the cavity, the digestive cavity that is produced by that inner sac. So here we go, we have the mesoglial mesoglial layer is right here. Remember, in the jellyfish, it's much thicker, so it's kind of a thin layer here. It does have sensory cells, so it does know a little bit about its surroundings, which is helpful when you have a nerve net to be able to pass that information along. Here is the cnidocyte. Now, Hydra do not sting their prey, but they can reach out and grab particles and bring them in. But just a very simple sac body plan. So kind of food in and back out. Oh, yeah, I guess I didn't really say, sorry about that. I was assuming you already know, this is our polyp phase, and it does contain a mouth and tentacles as well. So here's the Portuguese man-o-war, and the Portuguese man-o-war is a colony of polyps. So the mouth still faces upward, which doesn't look like it, I know. So this top polyp on a Portuguese man-o-war is filled with air, and that allows it to float at the surface. And then here are a whole bunch of other polyps that face in this direction. Now, if you know anything about Portuguese man-o-war, you know that they have cnidocytes and they contain toxins. Here obviously is the image of somebody who got stung by, looks like multiple polyps. Growing up in Fort Lauderdale, if we saw that air bubble on the top, we knew get out of the water because that is extremely painful. And these are not small colonies. They're usually quite large. So that is our Portuguese man-o-war, and they are the polyp form of Cnidaria. So what about our jellyfish? They fall into the class Scyphozoa. And just taking a look at it, remember the mouth on these points down. So here's the mouth. And they do have arms so that they can entrap their prey. Now, if you'll remember, the Hydra had a very thin mesoglial layer. The jellies have a very large, thick mesoglial layer, and you'll get to feel these in lab. It really does feel like jelly, kind of like a firm jelly. So they have a mouth, and then here's the stomach right here, and then they have gonads. There's usually 4 of them located on the underside. And again, you'll see those in lab as well. But when the mouth is pointing down, it is in the class Scyphozoa, and it is what's called the Medusa form. So these are some of our simplest animals. Now that we've started into the animal kingdom, we're going to continue going through the animal kingdom and look at some of our more and more complex phyla. But that is it for today with our invertebrates and, well, with these invertebrates. So we will see you next time.